Today we're going to talk about B cell development and selection. So remember that we're thinking about B cell development in the bone marrow. This is all coming from the hematopoietic stem cell, which first gets a signal and sort of chooses to either become a myeloid precursor or a lymphoid precursor. So we're in the sort of lymphoid precursor process, and we're in the bone marrow, and you should think about the bone marrow as sort of the antigen independent phase. What you can see here is that there are a few steps during from where the hematopoietic stem cell kind of commits to becoming a B cell. And we're really going to start thinking about this process with sort of this cell that has decided it's going to become a B cell. So this cell has already, become a lymphoid progenitor and has started to be a B cell. And as we talk through this, we're going to see we're going to call out some specific cell types in the process of becoming a B cell. And we're going to talk about events that those cells are doing, as well as things that are on the surface of those B cells. And when I say things that are on the surface of those B cells, I really mean B cell receptor parts or B cell receptor type things, because we're talking about making a B cell receptor. You can see that when we are looking at some of these different types of precursor cells, you can see the word progenitor is often used. So you can see we've got progenitors listed here. And the first cell that we're really going to think about is this cell that's called the pro-B cell. And so you can see the pro-B cell listed here. And so if you wanted to think about where the heck we got pro from, you might think about progenitor. So I want you to look at what we see in this image about the pro-B cell. And so each type of cell that we're going to talk about is sort of doing something. So it's got some events that it's going to be doing. And we're going to think about what it has in terms of its B cell receptor on the surface of the cell. So if you look at this pro-B cell here, you can see some proteins listed on the surface of the cell of the pro-B cell. Are any of those proteins labeled as the B cell receptor or anything that has to do with B cell receptor? So we've got some proteins, but none of them are the B cell receptor. They're other stuff. And so one of the key things about the pro-B cell is that it doesn't have any parts of the B cell receptor on its surface. And you will see that that sort of makes sense as we think through this process, because you will see that, well, the pro-B cell is actually doing some steps of making that. So if it hasn't made it yet, how can it have it on its surface? And specifically, the pro-B cell is doing two events. So sometimes we can divide the pro-B cell into like the early pro-B cell and the late pro-B cell based on which event it's doing. But really, I want us to just think about two events that the pro-B cell is doing. And so the first event that the pro-B cell does is rearrange a D segment in the heavy chain locus to a J segment. So we're putting a D with a J on the heavy chain. And then the second thing that the pro-B cell does is it puts a V with the DJ that it has made. You can see all of this on this slide as well. So here is my pro-B cell. At no point on this pro-B cell do you see B cell receptor on the surface. So no B cell receptor on the surface. And what you can see is that the first thing that this cell is doing is it's going from D to J of the heavy chain, and then it's going to put V with DJ of a heavy chain. Once we've done this recombination and put together a V, a D, and a J from the heavy chain, then we're going to test that heavy chain. Because why keep going with this process if the heavy chain you made failed, if the heavy chain you made is crappy? And so here you can sort of see this process as well. We're going to have DJ rearrangement of the heavy chain. Then we're going to have V to DJ rearrangement. And then we're going to put this heavy chain on the surface of the B cell and see if it works. See if we can get any kind of signals through this protein that we've hopefully made. Let's see how I have this set up. Okay, we'll go that way. So let's imagine first that it works. We are one of these B cells who made a good heavy chain. Yay us. Now we have that heavy chain on the surface. But we got a little bit of a problem if we want to put that heavy chain on the surface. Because are we actually putting a B cell receptor on the surface at this point? So I see a couple no head shakes. Why not? It might have CD79, but there's something else it still doesn't have. What's the receptor? Yeah. The light chain. We've only made a heavy chain. We haven't even made a light chain yet. And the B cell receptor, or the antibody, that protein that has two heavy chains and two light chains, doesn't really fold completely until it has a light chain. We haven't made a light chain yet. So we want to test this heavy chain and see if it's good, but we haven't made a light chain yet, and we can't fold our heavy chain correctly without a light chain. So let's imagine that you actually were someone who had this problem. What might you do if you wanted to test your heavy chain, but you didn't have a light chain yet, and you actually wanted to test the heavy chain, see that it was like a good heavy chain before you went through all the work of making a light chain? Turns out that what the B cell does is it has a fake light chain for this process. So usually the B cell receptor contains a heavy chain and a light chain. However, developing B cells make this other protein called surrogate light chain that can be paired with the heavy chain and sort of allow it to fold, allow it to sort of be like a B cell receptor, even though the light chain is not yet made. The surrogate light chain is made up of two different proteins. Their names are V3B and lambda 5. I have no idea why they were named that. They don't vary. So when we eventually get a B cell receptor with the heavy chain and the light chain, every B cell is going to, have that different B cell receptor. Here, everybody uses the same surrogate light chain. It's really testing, could we get a functional heavy chain on this B cell? Did we actually make a B cell receptor that works? And now, if you think about it, we've changed something in my table. We don't have nothing on the surface anymore. Now we've got something on the surface. Specifically, we have the heavy chain and the surrogate light chain. Which is V3B and lambda 5. This combination of the heavy chain and the surrogate light chain is known as the pre-B cell receptor. Sometimes known as the pre-BCR. And so you can see the pre-BCR here, and you can see how it compares to a regular B cell receptor. And what you might notice, Brenna, is you actually do have CD79A and B here. I just hadn't really been mentioning. It was there before, yeah. The B cell starts making it even before it starts making heavy and light chain.
And so that's why you mentioned that it might be missing the intracellular part.
It's not, exactly. But now, this cell that is testing this heavy chain has something different on its surface, so it no longer qualifies as a pro-B cell. It is now the next cell in this list. It now qualifies as something else. And the something else that it qualifies as is called the pre-B cell. So the pre-B cell has the pre-B cell receptor. And so now when we have the pre-B cell receptor on the surface of the cell, we call this the pre-B cell. And pre-B cells are going to do, again, a bunch of functions. So we're going to see a bunch of different things happening with the pre-B cell that we can think a little bit about. But a lot of those functions of the pre-B cell are really based on whether or not the pre-B cell gets a signal through the pre-B cell receptor. And so we have to think a little bit about what's going on with the pre-B cell receptor and how it might get a signal. Because the idea with the pre-B cell, and the pre-B cell receptor is that basically we made this heavy chain as a pro-B cell. Now we put it on the cell surface and we're trying to test it to see if it worked. And so we ideally hoping to get a signal through this receptor. If you recall, the B cell receptor or the antibody binds to antigen using the combination of the heavy chain and the light chain, right? It needs contacts with both heavy chain and light chain to contact antigen. Here, we don't have a light chain yet. We have this non-varying surrogate light chain thing. So how the heck could we get a signal? And the way that the pre-B cell receptor signals is shown here. So basically, you can see that the surrogate light chain has these like little tails on the end of it. And the surrogate light chain will basically just bind to another surrogate light chain on the surface of another B cell receptor and pull a bunch of B cell receptors together. So we don't need an antigen. We just need to pull together some of these receptors that have good heavy chains, and we get a signal. So one of the things we probably should think about is, I keep saying that we're testing to see if we have a good heavy chain. A good heavy chain. We should think about what's a good heavy chain. What makes a heavy chain a good heavy chain? Or more usefully, what makes a heavy chain a bad heavy chain? Because we're testing to see if we made a good heavy chain. And if we think about what makes a good heavy chain, some of this will make a little more sense. So remember that in order to make that heavy chain, we had to put together a B, a D, and a J segment, right? And then we also had the addition of P and N nucleotides. We also maybe had some exonucleolytic cleavage. So let's imagine that when I added my P and N nucleotides, I added in total, in making this heavy chain, 5 base pairs. There's a potential problem that could happen when I added 5 base pairs. What problem might happen when I add 5 base pairs? It also could happen if I added 4 base pairs, but would not happen if I added 6. Yeah? Mm-hmm. Yeah, so we'll say we have got some V, we've got some D, we've got some J, we've got some constant region. We want to translate this as one big long protein, right? And we even need a transmembrane domain so that we can be a receptor. We need to be able to translate this whole one long protein. In a perfect world, there should be a stop codon right here, right? Where you stop translation and stop making the protein. If you add something other than a multiple of 3 base pairs, you're going to shift the reading frame further down, and you might insert a stop codon. So now you might put a stop codon there. You might get a protein that has a beautiful V, D, and J, but no constant region, no transmembrane protein. And so it actually can't ever be a heavy chain that looks like this. It's just like this part of it and then a stop codon never is going to have a transmembrane domain, never is going to be a good heavy chain. It's just going to be a sucky heavy chain. And so the most common reason why we would have a bad heavy chain, or why this sort of process would fail, is that if we have a frame shift. So if we switch the reading frame, as Brian was mentioning. And so if we do that, we don't even get a protein that is a full heavy chain that makes anything like this. So really what we're testing for is did you get a heavy chain that actually has all the parts and that can bind to a surrogate light chain? We don't really care what antigen it binds to. We don't really care any of that. It's did you competently make a heavy chain? Is really what we're testing for here. Any old heavy chain can bind to surrogate light chain. Once you have your heavy chain and surrogate light chain bound, has a transmembrane domain, has all the parts, then we'll get a signal in this B cell and be like, okay, B cell, keep going. You did a good job. You get a pat on the head. You may keep trying in this process. You didn't even make a protein of a heavy chain. You were that incompetent. We don't want you to keep going and try to make a light chain when you don't have a heavy chain to pair it with. That would be a waste of time. And so we're going to be doing this signaling. We're going to have this pre-B cell receptor. We're going to be getting this signal. And really what we're signaling for is, did you competently make a heavy chain? And what you can notice here is that this shows that only about half the cells actually competently make that process. And we'll talk about what happens to the cells that didn't competently make that heavy chain in a bit. So once we have the pre-B cell receptor on the surface of the cell, and we're successfully getting a signal through that pre-B cell receptor, we're going to see a number of different signals sort of be given to that cell. So that cell is going to get a bunch of different things. If I really wanted to, I could divide up the pre-B cell into like multiple things about different events that we're going to see. Some of them are pretty important and pretty easy. Some of them get a little trickier. So the first one I want you to notice is I'm actually going to say something about the thing that is there as number 2, is that we're going to turn off RAG. So we're going to stop making RAG1 and RAG2 for a bit. This has a couple of important reasons. I'm only going to tell you one right now. I'm going to tell you the second one in like a slide or two. So now the cell has competently made a heavy chain. Yay! What we would like it to do long term is make a light chain, right? And finish this process. In order to finish this process, remember I told you that developing lymphocytes are really needy and they need a lot of pats on the head and to be told that they did a good job. And so this signal is a pat on the head. And so one of the things that the cell's also going to do is it's going to survive. So we're going to get a survival signal. We're going to tell the cell, yes, you should live and keep going with this process. And we're also going to tell the cell one other key thing about this, which is that we're going to tell the cell to divide. So we're going to tell the cell to proliferate, which is just a fancy word for divide. Because ideally, now that we have this one really good heavy chain that was made, we'd like to make many copies of that cell so that each of those new progenies could make a different light chain. And we can get lots of combos of light chains to go with this one good heavy chain we made. So if we know you made a good heavy chain, then we want you to make a bunch of copies of yourself so that we can pair that good heavy chain with multiple light chains. And one thing I want to mention is it's actually really important that we've turned off RAG here, because we do not want RAG1 and RAG2 on if the cell is going to proliferate. We do not want any risk of DNA damage when we're actually going to replicate the DNA and go through the cell cycle. So we make sure RAG is completely off so that there's no DNA damage that happens during this proliferation. So we're also going to do another thing that is really important here, which is that we're going to stop making, and really when I say making, I mean transcribing, surrogate light chain. You don't need to synthesize more surrogate light chain, because in theory, you're not going to need it anymore, because I know this is a big spoiler, we're going to make a light chain next. So you don't need a fake light chain anymore. You can stop synthesizing fake light chain because it's time to make the real light chain. And we will eventually turn RAG back on and do our light chain recombination. So all of these things are events that happen in the pre-B cell. And all of them are, and I kind of have them in order, all of them happen if the cell gets a signal from the pre-B cell receptor. Like basically the cell is told, you made a heavy chain correctly. You did it right. Now you are allowed to do all of these events. And you can see sort of this whole process here as well. And what I want you to notice is here's our pro B cell. It has RAG on. It does not have a B cell receptor on its surface. Now we get to a pre-B cell where we have this pre-B cell receptor on the surface. And we have, at least at first, turned off RAG. Turning off RAG at the beginning has one other reason why it is important. And to do this, to fully explain this, I'm going to get to do some drawing now. This is why I was like paying attention to the chalk, because I need to make sure I have a bunch of chalk colors to do this drawing. All right, this should work. My elementary school teachers used to yell at me for always drawing everything like on a slant. I'm clearly not grown out of that. Maybe it's because I'm small. I draw to the point where I can't reach anymore too easily. Because I can't reach. Okay, so you can imagine this rearranging, right? We're gonna have to pick one of my V's, put it with a J, pick one of the D or the B's, put it with the DJ. You can imagine all this happening, right? Which Shelby, which V, which D, and which J would you like to pick? Okay. 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 So there's a little bit of a problem, however, in our organism or in our cell. to draw these in two different colors. What am I trying to signify here with the colors? And I use specific colors on purpose. It's not that it's double-stranded, but you're getting there. Nope. What colors did I use? Pink and blue. What colors go with pink and blue? Or what goes with pink and blue? What do you think of when you think of pink and blue? What? Okay. I don't. Okay, so our cotton candy is either a girl or a boy. So why did I make two versions of this, a girl and a boy? Because you have two copies of this chromosome in every one of your cells. One that you got from your mom and one that you got from your dad. So if we make this rearrangement that shall be picked out on mom's chromosome, that's cool. Brian, what one would you pick? And if the other chromosome does that rearrangement, we now have a B cell that's making two heavy chains that are specific potentially for two antigens. That's not how this works. So we can't really have both chromosomes rearranged. Either mom's chromosome or dad's chromosome has to rearrange. And if you do the other one too, then terrible death and destruction happens. Specifically, you get a B cell that's responding to two things. And so we actually have to make sure that this process doesn't happen. And so when we turn off RAG1 at this step of pre-B cell, in the pre-B cell, we often also refer to this as allelic exclusion. Because we are turning off RAG1 and RAG2's ability exclusion to make heavy chains or to recombine heavy chain. So the basic idea here is as soon as one chromosome, it could be mom's chromosome or dad's chromosome, finishes, soon as it's done, we get the protein on the surface of the cell. If that protein worked, if we get a signal, then that signal says, nope, can't make more heavy chains. Sorry, dad's chromosome. You're not allowed to finish. Stop. And this allele, this second chromosome, could be whichever chromosome, the one that didn't finish yet is excluded from being able to keep going in this process. And so when we're turning off RAG here, we're very specifically turning off the ability to make any further heavy chains so that we can do allelic exclusion and make sure that whichever that was that first heavy chain that was made is the only one we make, and we don't make any more. And so if you look way back here, you can see in reality, when we do the first step in the pro-B cell and we're doing D to J, both chromosomes tend to do D to J. But then somebody wins in terms of getting the V to DJ done first. And as soon as that one's done, we try to put it on the surface and see if it worked. And if it worked, we stop the other one. You can imagine if it didn't work, then we don't stop the other one. But if it worked, we stop the other one. We're like, nope, we do not want to have two receptors. Yes. We're going to get there. Yes. Great question of what if neither of them worked? We're coming to that. I put this slide in, but because of how I drew stuff on the board, I don't need this slide. And so this process is really, like I said, allowing us for allelic exclusion. This is sort of trying to show us why allelic exclusion is important, and I'm going to make another little drawing to show us about importance of allelic exclusion as well. So because we do allelic exclusion, and we are in fact going to do allelic exclusion at both the heavy chain and the light chain, we get one B cell that only makes a bunch of copies of the same B cell receptor. Here you can see it's blue heavy chain, red light chain. If we didn't do that, then we could make more than one heavy chain. So you can see there's blue and green, like one from mom and one from dad's chromosome. And you could make like asymmetric pairings, and you could make all these different light chains, and you'd see that we can't bind antigen very tightly, like we could over here, because we have all these different wacky receptors. And even more importantly, let's imagine that this receptor binds to antigens from SARS-CoV-2. Do you want this receptor? Do you want to make antibodies against SARS-CoV-2? Yes, because you would like to be protected against that virus, right? You want to give that B cell all the pats on the head and be like, yes, I like you a lot. Let's imagine this receptor binds to myelin in the myelin sheath of your brain. Do you want this one? No. So I want this B cell to not get a pat on the head. It would be really bad if it's the same B cell. And so it's actually really important that each B cell is specific to one thing, and then you can do your selection on, do I like this one thing or not? If we didn't have allelic exclusion, then like I said, you'd have this bad affinity, which is shown here, crappy binding strength, but you'd also have this just huge mix-up of what things you could bind. And so imagine, if you will, that we have no allelic exclusion. You could make heavy chain from mom. You could make heavy chain from dad. And you could put them with mom's kappa. You could put them with dad's kappa. You can put them with mom's lambda. You can put them with dad's lambda. And you could even also have the versions where it's not symmetric, where it like mixes and matches like this one, but that's too much drawing for me. And so you could end up with a huge number of receptors on the surface of the cell if we don't have allelic exclusion. And so this early turning off of RAG, and specifically RAG's ability to access the heavy chain, is super important in that pre-B cell. And so this is sort of the overview of sort of that testing that's going on with these cells. As we sort of hinted at previously, it's also possible that when we put our attempt at a heavy chain on the surface, it failed. Maybe we put a stop code on it and we didn't get a full protein. Maybe we had some issue. Well, fortunately, because we did not ever get a good signal, we did not ever turn off RAG. And so now the second chromosome can keep going and try to recombine. So we got 2 chances to make a good heavy chain. If that one works, oh my gosh, it goes on the surface and gets a signal and survives. But if that one doesn't work, this gets to your question, Shelby, what if we fail on both chromosomes? The cell dies. It dies by apoptosis because it never gets a pat on the head that said it did a good job. It used up its 2 chances of making a heavy chain. It was a failure of a pro B cell, and it dies and doesn't get to go on to the next part of its life because it never successfully made a heavy chain. And you can see another version of that here. First, we do all the D to J's of the heavy chain. We know it's heavy chain because there's an H. Then the first chromosome finishes V to DJ. Maybe it made a good pre-B cell receptor. Maybe it failed, so the second chromosome tried. Maybe it worked. Yay. Maybe it failed. If it failed, then the cell dies because it can't get a signal. Yeah. So if you made a successful protein, you can get a signal through that protein. And so then you turn off RAG because you don't need it anymore because you were successful. Yeah.
Why would it die instead of just like try again?
What's it going to try again?
Well, I thought the reason it failed because of like
It is completely random.
So it's just like.
So how's it going to try again? Remember, this is a heavy chain. So when we pick our V, D, and J, we've gotten rid of the intervening stuff, including RSS. You can't try again, right? There are no more D's to like try this again. So, same thing here. Well, those would still be there. But you can't try again. There's no ability to try again. So this cell has already sort of like specialized to B cell life. It's not going to like become a skin cell now. It's sort of specialized to try to be a B cell, and it failed being a B cell. So sorry, B cell, no more ATPs for you. Yes. Mm-hmm. We do not want that situation where one B cell makes all of those. We would love it if it turns out that 8 B cells, can I count? Yeah, 8 B cells, each one of those were made. We'd love it for the population, but we do not want one B cell making all 8, because that would be very confusing for that one B cell. Yeah, absolutely. We use both of them at some frequency. It's totally random which one gets used first, which one gets used second. So genetically, you use them all, but each individual B cell uses just one. Yeah. And what you'll notice here, just to sort of get at that point, like half of the cells that start this process die just because they can't make a heavy chain. So the immune system, as you will see throughout the entire semester, is not a model of efficiency. We will have places where like 90-something% of cells die often, but we have selected evolutionarily for these processes because we get breadth. So after we've had this early stage in our pre-B cell where RAG was off, and we've been able to do things like allelically exclude, survive, proliferate, all that good stuff. We kind of got told we were good at being a pre-B cell. Now we can turn RAG back on. And when we turn RAG back on, RAG is not going to the heavy chain anymore. RAG is now going to go to the light chain and make light chain rearrangements. And so you can see the sort of final thing that our pre-B cell is doing is putting RAG back on, and it's doing rearrangement of the light chain, B to J of L, B to J of the light chain. Once we have made that light chain, we put the light chain on the surface because we want to test it, just like we needed to test the heavy chain. And I need a bigger chalkboard or smaller handwriting. But now I'm going to have a different group of cells with something different on their surface. And now the thing on the surface is the B cell receptor. Actual B cell receptor. Actual heavy chain plus actual light chain. Now that there's something different on the surface of the cell, it doesn't get called a pre-B cell anymore. It gets a new name. And the new name for this cell is an immature B cell. Yeah.
I thought we were talking about different types of B cells, or we were talking about the B cell lifespan.
We're talking about like different stages in the B cell's life. So like first it's a pro B cell, then it grows up and becomes a pre B cell, then it grows up and becomes an immature B cell. Yes. And so our final cell type is the immature B cell. Now it's got an actual immunoglobulin molecule on its surface. And it's going to do some additional types of testing of the BCR that are known as central tolerance mechanisms or central tolerance testing. And right now I'm just going to say central tolerance and we'll see where I get it. Actually, I talk about central tolerance. And so, again, if everything works out perfectly, we now are going to have our immature B cell. It's going to have the B cell receptor on its surface, and it's going to get to leave the bone marrow and go out to the rest of the body and search for antigen. So if everything works perfectly, if we're happily ever after, our immature B cell, it gets to leave, gets to go out of the bone marrow, it finally gets to like go into the real world and try to find antigens. But of course, just like we did with the heavy chain, we need to test the light chain. The sort of early way of thinking about light chain testing, you can think of as being kind of similar to what we were thinking about with the heavy chain. Did you competently make a light chain? Did you make a protein that was in frame? Or did you add stop codons? With the light chain, it turns out that we have multiple light chain gene segments in the genome, or multiple light chain loci in the genome. We have one called kappa, we have another one called lambda, and they are on different chromosomes. Literally the only thing I know about kappa and lambda is that when B cells do this process, they try kappa before they try lambda. So they try both kappas. So they try to get a signal. If they get a signal, woohoo, I'm an immature B cell. If they fail, they try the other kappa. If it works, woohoo. They fail, then they try the first lambda. That fails, they try the second lambda. So they get 4 chances on light chains, while they only had 2 chances on heavy chains. And if they fail all 4 light chains, then they're going to die. They try kappa. Kappa's the K. They try kappa first. And then after they try kappa, they try lambda. Literally, we don't know anything about functional differences. We don't know any other reason why we care. We do know, however, that if we see a B cell that uses lambda, it must have failed at least twice. Yeah, that's lambda. Mu. That's the mu. Also, if you go on Moodle, there's a thing that says it's like a Greek letter reference guide, and it has all the Greek letters, both I think it has capital and lowercase, and says their name, and says how to type it, if you need to. Huh?
Like U-E-M?
U is M.
Like U.
But it goes with M. Who knew that when I was in a sorority learning all this stuff in college, I was actually studying immunology? And so here's the version of this from your textbook. You can see that in our pro B cell, we're doing D to J on both chromosomes. Then we're trying to do B to DJ of the heavy chain. Once we get a successful heavy chain, we are now called a pre-B cell. Now we have 4 different tries to make a successful light chain and become an immature B cell. We first get to try our 2 kappas. We then get to try our 2 lambdas. If we fail all of that, then we undergo apoptosis. One other super important thing to know about immature B cells, and we're going to imagine that all the testing goes well. We're going to imagine that we are an immature B cell, everything goes well, and you get to leave. When you leave the bone marrow as this immature B cell, you never make RAG again. Once you turn RAG off at this point, once you get a good signal here from that B cell receptor, you turn off RAG again. You also, this might shock you, survive and proliferate. You never are going to turn RAG on ever again for the rest of your life because you did all this hard work to make a good piece of DNA. You are never putting it back on. You do not want to mess up your DNA now that you've done all this hard work. One other thing that you can notice is when these B cells are just coming out of the bone marrow, you can see it's showing them making sort of a black B cell receptor and also a red one, kind of right as they're coming out. That's because they're making both IgM and IgD. So recall that IgM and IgD come from the same RNA transcript. So at the beginning of doing transcription at this locus, we actually include both the IgM and the IgD sections and can switch between them by splicing the DNA. And so these B cells that have just come out of the bone marrow are usually have both IgM and IgD on their surface. In my experience, the only reason I've ever cared about IgD is to help me know if a B cell is new and just came out of the bone marrow or not. Because B cells that have just come out of the bone marrow are making both M and D. That's legitimately the only function I originally learned for IgD. And this whole process that we are seeing here, I want you to remember was in those primary lymphoid organs, where we don't have a lot of foreign antigens. And this is all happening before we let those cells go out into the real world and circulate throughout the body. And I've shown you this slide before. But when I've shown you this slide before, which is this exact version of the slide, I actually have had some white boxes over some stuff in oversimplifying what really happens. And you guys actually did ask a great question about this at the time. We've been thinking about VDJ recombination where we're making this massive diversity of the receptors. We're making every single receptor possible. Every combo of V's, D's, and J's, and all the added nucleotide, every single one possible, right? Some of those will bind to self-antigen. Some of those actually would give you autoimmune disease. And so before we let the B cells out of the primary lymphoid organ into the real world, we actually run this one last test on those immature B cells and check and see, do those B cells bind to self-antigen? If those B cells bind to self-antigen, then it would be really bad to let them go out into the real world and respond to antigen, because then they're going to respond to whatever self-antigen there is, and they're going to give you an autoimmune disease. And so we make this massive diversity, and then we do this testing process where we test for those that respond to self-antigens, and we delete them. We actually kill those cells. So they are not allowed to leave the bone marrow. They can't induce an autoimmune reaction. And we only let the ones that make it through this testing process leave the bone marrow. Self-antigen being like no, any part of your body, like actin, myelin, like every protein that's part of your body, all of the U's. So this process is known as thinking about something called central tolerance. Immunologists use the word tolerance to mean reasons why you don't make a really bad immune response to yourself, or reasons why you avoid reacting to self proteins, lipids, nucleic acids, etc. So you can think of tolerance as sort of like how we avoid autoimmunity, how we avoid responding to self antigens. When we think about tolerance mechanisms, there are two kinds of tolerance mechanisms. One of them is called central tolerance, which you can see here. Central tolerance means a tolerance mechanism that happens during development of the cell in a primary lymphoid organ. So central tolerance means it's a kind of tolerance mechanism that happens in primary lymphoid organs. Later on, we will also see a mechanism called peripheral tolerance. Peripheral tolerance is when you have a tolerance mechanism that happens after the primary lymphoid organ, after the cell develops. So peripheral tolerance is sort of a later, what happens in the real world to the cell, while central tolerance is what happens earlier on. And so to give a little bit of an overview of the central tolerance mechanisms, if our developing B cell, our immature B cell, does not react with any antigens in the bone marrow, then it moves out to the blood, it leaves the bone marrow, makes IgM and IgD, and lives happily ever after. But if our immature B cell does recognize some kind of self-antigen, then it gets held up in the bone marrow, and we got to do something about it. Because we don't want to release that cell out into the rest of the body to potentially cause issues. That's where I'm going to leave us today. On Friday, we're going to talk about the Ottinger paper. Depending on timing, I may finish up what I was going to say about central tolerance, or I may leave it till after the exam. We'll see. Basically, the last thing I was going to tell you for the exam is there's 3 ways central tolerance happens for B cells. And I was going to tell it to you with like showing you the experiments that proved it, but I might just say, here's the 3 ways and not tell you the experiments. So we might do that on Friday, just to know, like, here are the 3 ways. That's all I wanted to get through before the exam. So they are pretty cool. Well, don't worry, we'll get there. But right now, we've got enough. So I'll see you guys tomorrow, and I'll have handouts for you. Remember your problem set by the end of the day, and remember your lab stuff if you didn't already give it to me.